Dear students, today we see hormones and health production of hormones as pharmaceuticals. Introduction. In its modern conception, biotechnology is the use of genetic engineering techniques to manipulate microorganisms, plants and animals in order to produce commercial products and processes that benefit man. These techniques, which are the backbone of the biotechnological revolution that began in the mid-1970s, have permitted the isolation and manipulation of specific genes and the development of transgenic microorganisms that produce mainly eukaryotic proteins of therapeutic use such as hormones, enzymes and vaccines. Biotechnology is present in diverse areas such as food production, degradation of industrial waste, mining and medicine. Recent achievements include drug production in transgenic animals and plants, as well as the commercial exploitation of gene sequences generated by the Human Genome Project and similar projects of plants and animals of commercial interest that are and will be in process. Human growth hormone was, after insulin, the second product of the new technology. Growth hormones from different animal species have also been produced in transgenic organisms, and these have been used in different examples in aquatic animal and livestock sectors. Types of biotech pharmaceuticals. Many biotech pharmaceuticals are similar or identical to proteins that healthy human bodies produce routinely for normal functions. In addition to gene therapy drugs, following are the major types. Number 1. Hormones. Recombinant human insulin became the first manufactured or commercial recombinant pharmaceutical in 1982 when the FDA approved human insulin for the treatment of cases of diabetes that required the hormone. Before the development of recombinant human insulin, animals, notably pigs and cattle, were the only non-human sources of insulin. Animal insulin, however, differs slightly but significantly from human insulin and elicit troublesome immune responses. The therapeutic effects of recombinant human insulin in humans are identical to those of porcine insulin, and it acts as quickly as porcine insulin, but its immune system side effects are relatively infrequent. Further, it can satisfy medical needs more readily and more affordably. Other recombinant hormones include those described below. Lispro. Regular insulin ordinarily must be injected 30 to 45 minutes before meals to control blood sugar levels. Lispro, that is human log, a recombinant insulin-like substance, is faster acting than regular insulin. Injection of Lispro is appropriate within 15 minutes before meals. Using it instead of regular insulin may be more convenient for some patients. Epoetin alpha. Erythropoietin, that is EPO, a hormone produced by the kidneys, stimulates the bone marrow to produce red blood cells. The FDA has approved recombinant EPO, epoetin alpha, for the treatment of anemia due to chronic renal failure. Recombinant human growth hormone. Human growth hormone HGH is used to counter growth failure in children, that is due to lack of HGH production by the body. Before the introduction of recombinant HGH, the hormone was derived from human cadavers. Cadaver-derived HGH was susceptible to contamination with slow viruses that attack nerve tissue. Such infective agents caused fatal illness in some patients. Recombinant HGH has greatly improved the long-term treatment of children whose bodies do not produce enough HGH. Number 2, cytokines. Cytokines are hormone-like molecules that can control reactions between cells. They active immune system cells such as lymphocytes and macrophages. Cytokines that have recombinant variants or versions include those described below. Interferons are potent cytokines that act against viruses and uncontrolled cell proliferation, which is the primary hallmark of cancer. Virtually all conventional chemotherapeutic agents act directly on cancer cells. When interferons act on cancer cells, however, they do so indirectly by affecting the functioning of the immune system. The FDA has approved certain recombinant interferons for the treatment of several diseases, including AIDS-related Kaposis sarcoma, hair cell leukemia, hepatitis B, and genital warts. Interleukins function as messengers between leukocytes. Interleukin 2, that is IL-2, stimulates T lymphocytes. The FDA has approved a recombinant variant of IL-2, aldesleukin, that is proleukin, for treating renal cell carcinoma. The anti-tumor effect of IL-2 and its recombinant variant is directly proportional to how much of the agent is administered. Endogenous IL-2 is scarce. Aldesleukin can be mass produced but has adverse side effects at relatively low levels of administration. Granulocyte colony stimulating factor, that is GCSF, stimulates the bone marrow to produce neutrophils, that is antibacterial leukocytes. The FDA has approved a recombinant variant of GCSF, filgrastim, for controlling infections in patients on anti-cancer drugs that suppress immune responses, in patients undergoing bone marrow transplantation, and in patients with neutropenia. GM-CSF stimulates the bone marrow to produce neutrophils and macrophages. The FDA has approved its recombinant equivalent sargramostin, that is leucine, for administration to cancer patients who destroyed their bone marrow because of intensive chemo and or radiotherapy and have undergone a transplant. Sargramostin is administered until the transplanted marrow can produce leukocytes adequately without such stimulation. By keeping leukocyte levels high enough to control infections, sargramostin can hasten recovery. Number 3. Enzymes. Below are descriptions of recombinant enzymes and diseases against which they are effective. Alteplase. The process of dissolving blood clot in circulatory system involves conversion of the protein plasminogen to the proteolytic enzyme plasmin. A recombinant version of one of the enzymes that accelerates the conversion can contribute to the treatment of heart attacks, strokes, and pulmonary emboli. This recombinant enzyme is recombinant tissue type plasminogen activator, that is Alteplase. The effects of Alteplase are more localized than those of other enzymes used to dissolve blood clots, that is streptokinase and urokinase. Thus, in theory, Alteplase would cause less bleeding throughout the body. DNase alpha. Cystic fibrosis, that is CF, is a genetic disorder marked by excessive mucus secretions and frequent lung infections. About half of those with cystic fibrosis live fewer than 29 years. A DNA splitting enzyme produced by the body Deoxyribonuclease I, that is DNase I, can break down DNA that is outside cells but not DNA that is within intact cells. In contrast, DNase alpha plumozyme, a recombinant variant of DNase I in aerosol form, breaks down intracellular DNA. Decomposition of the intracellular DNA in the excessive mucus secretion that dispose person with CF to lung infections can make the secretion less adhesive to airways. DNase alpha can thus decrease the incidence and duration of both lung infections and hospital stays in CF patients. It is the first new drug the FDA has approved in 30 years for the management of CF. Imiglucerase. Gaucher disease, characteristic by bone destruction and enlargement of the liver and spleen, is due to an hereditary deficiency of glucocerebrosidase. A variant of this enzyme is obtained from human placentas, but 20,000 placentas would provide only a year's supply for a single patient, and everyone with the disease has a lifelong need for such an enzyme. The FDA has approved a recombinant variant of glucocerebrosidase, imiglucerase, that should end the supply problem. Number 4. Clotting factors. Inadequate bodily synthesis of any of the many clotting factors can prevent effective clotting. The FDA has approved 2 clotting related recombinant drugs, Abcimab for the prevention of blood clotting as an adjunct to angioplasty and recombinant anti-hemophiliac factor RAHF for the treatment of hemophilia A. Before the introduction of RAHF, treatment of hemophilia A required protein concentrates from human plasma. Such concentrates could contain contaminants, for example HIV, and the lifetime treatment of single patient required thousands of blood contribution. Persons with hemophilia B lack factor IX. They require either factor IX concentrates from pooled human blood or factor IX from cell cultures, that is, some of which are genetically engineered. In July 1997, Scotland's Roslin Institute announced the birth of the first genetically engineered sheep clone. The clone carries a human gene for factor IX and it gives milk that contains the factor. Other multicellular organisms that have been genetically engineered to produce substances that are or may be medically useful to humans include cows, goats and rats, and corn, potato and tobacco plants. Number 5. Vaccines. In every modern vaccine, the main or sole active ingredient consists of killed microorganisms, non-virulent microorganisms, microbial products, for example toxins, or microbial components that have been purified. All these active ingredients are antigens, substances that can stimulate the immune system to produce specific antibodies. Such stimulation leaves the immune system prepared to destroy bacteria or viruses whose antigens correspond to the antibodies it has learned to produce. Although conventionally produced vaccines are generally harmless, some of them may rarely contain infectious contaminants. Vaccines whose active ingredients are recombinant antigens do not carry this slight risk. More than 350 million persons worldwide are infected with the virus that cause hepatitis B, a major cause of chronic inflammation of the liver, cirrhosis of the liver and liver cancer. The first hepatitis B vaccine available in the US was made with derivatives of plasma from person with chronic HBV infections. A recombinant vaccine whose sole active ingredient is a recombinant and thus uncontaminated antigen has replaced it. Use of this vaccine is very cost effective, especially in North America, since interferon treatment for hepatitis B is very expensive. The Ebola virus, first identified in 1976, causes Ebola hemorrhagic fever, one of the deadliest viral diseases known. About 50 to 90% of patients infected with the Ebola viruses consequently die. In 1997, American researchers announced that an experimental recombinant vaccine against the virus had proved effective in mice and guinea pigs. Because of immune system inadequacy, some groups, infants and young children, for example, tend to respond poorly to vaccination against certain bacterial infections, for example, streptococcal pneumonia. Preliminary research suggested that antibacterial vaccines that contain specific antibodies are more effective against such diseases than are comparable conventional vaccines which do not contain antibodies. Although vaccines traditionally have been designed to prevent only infectious diseases, the development of individualized vaccines made from the cancer cells of each patient to restrain, prevent the recurrence of or cure some forms of cancer is promising. Researchers at the US National Cancer Institute have demonstrated that a special vaccine plus interleukin-2 can shrink tumors in patients with metastatic melanoma. The vaccine used in this study contained a melanoma antigen variant more effective than the original antigen at attracting to cancer sites T lymphocytes that are destructive to tumors. Another prospect is effective inoculation by ingestion. In February 1998, US researchers announced that they had genetically engineered potatoes to produce a vaccine against cholera. Every year, 5 million people contract cholera and 2 lakh died from it. The vaccine is a non-toxic, relatively heat-stable protein that can elicit an immune response even when it is ingested as a potato constituent. Number 6. Monoclonal antibodies. All the antibodies the immune system normally produces in response to a specific antigen are capable of marking, that is, binding to that antigen, but these antibodies termed polyclonal are varied, not identical. Monoclonal antibodies, that is MoAbs, that share a specific antigenic target are identical and are more sensitive to that target than are polyclonal antibodies for the same antigen. MoAbs are the products of hybridomas cells that result from the Biotech fusion of bone marrow tumor cells and B lymphocytes. Hybridomas can be geared to produce specific MoAbs continuously. Theoretically, a MoAb and designed for a particular antigen on cancer cells can initiate an immune response that would destroy cancer cells without harming normal cells. At least 26 MoAbs are undergoing clinical testing as anti-cancer agents, but The medical potential of MoAbs extends to many other diseases. For example, the FDA has approved the MoAb drug MoAb CD3 for the treatment of immune system rejection of transplanted hearts, kidneys and livers. MoAb CD3 restrains immune response and thus increases the likelihood that the transplant will function. More recently, the FDA approved the immune suppressant daclizumab, that is Zinapax, for the prevention of kidney transplant rejection. Daclizumab active ingredient is the humanized MoAb. 90% of the MoAb's amino acid structure is human. Thus, the likelihood of an allergic reaction to it is low. Another MoAb, infliximab CA2, appears effective against Crohn's disease, an immunosystem disorder marked by intestinal inflammation. Infliximab is specific for a factor in the development of the disease. The medical utility of MoAbs is not limited to therapeutics. Because of their ability to bind to specific antigens, MoAbs have been used for many years to identify antigen carrying disease agents than to locate them in the human body. Recently, British researchers designed MoAbs that may be useful in determining whether cancer has spread from breast tissue to axillary lymph nodes. The spread of cancer to other parts of the body is likelier if the cancer has spread to lymph nodes than if it has not. Traditionally, determining whether the lymph nodes have been affected involves surgery, but using radio-labeled MoAbs specific to antigens on malignant cells enables locating such cells with an instrument comparable to a Geiger counter and may decrease the need for surgery. The ability of MoAbs to bind to and thus tag specific proteins also makes them potentially useful in the diagnostic imaging of internal organs and tumors. Other biotech drugs listed below are a few of hundreds of other biotech drugs that are either in clinical use or undergoing scientific investigation. Biotech vaccines undergoing investigation include vaccines for acellular pertussis, that is whooping cough, AIDS, herpes simplex, Lyme disease and melanoma. 2 new recombinant interferons are undergoing investigation, consensus interferon for treating hepatitis C and recombinant beta interferon 1A for multiple sclerosis. Recombinant protein tyrosine kinase, that is PTK, inhibitors may have therapeutic utility against diseases marked by cell proliferation such as cancer and atherosclerosis. Protein tyrosine kinases contribute to cell division and are the targets of these biotech drugs. Recombinant human interleukin 3 is undergoing clinical investigation as an adjunct to traditional cancer chemotherapy. 2 recombinant growth factors, that is cytokines, that regulate cell division are undergoing major clinical trials. Recombinant human insulin-like growth factor RHIGF1 and recombinant human platelet-derived growth factor, that is PDGF. Injecting the recombinant protein fibroblast growth factor FGF1 into the human myocardium increases the blood supply to the heart by introducing blood vessel formation. Injecting the recombinant protein fibroblast growth factor FGF1 into the human myocardium increases the blood supply to the heart by inducing blood vessel formation. Such treatment called a biological bypass or biobypass that does not require surgery. FGF1 is injectable non-surgically into the myocardium by cardiac catheterization. A biobypass may benefit person with coronary artery diseases whose arteries are not repairable surgically. There is gene therapy for of biopass, that is VEGF gene therapy. In January 1998, advisors to the FDA recommended that the agency approved Apligraf, a recombinant skin replacer for the treatment of leg ulcers due to poor circulation, and Dermagraft, another such product for the treatment of diabetic ulcers. Traditionally, patients with chronic skin ulcers or severe burns have had only two treatment options: skin grafts, which depended on how much healthy skin they had, and temporary protective coverings made of dead cells. The FDA approved Apligraf in May 1998. At the end, to conclude, virtually all biotherapeutic agents in clinical use are biotech pharmaceuticals. A biotech pharmaceutical is simply any medically useful drug whose manufacture involves microorganisms or substances that living organisms produce, for example, hormones, enzymes, etc. Most biotech pharmaceuticals are recombinant, that is, produced by genetic engineering. Insulin was amongst the earliest recombinant drug. Biotechnology encompasses such disparate processes as industrial fermentation, gene therapy, and cloning. The medical repercussions of advances in biotech have been impressive, but the implications of those advances for human health are no less than staggering. Thank you.